Chapter 6 is going to be a more in-depth look into protein. Proteins are a little bit different than the other nutrients because they contain one additional atom, which is nitrogen. They're made of carbon, hydrogen, oxygen, and then also nitrogen. The building block that proteins are made of is called amino acids. And here you see a picture of what an amino acid is composed of. So each amino acid has a central carbon that has a hydrogen attached to it. It also has an acid group attached to it and what we call the amine or amino group. And then at the top, you see this blank box called the side group or the side chain. Now, the reason that this is blank is because this is different for each individual amino acid. We actually have 20 amino acids, and each one of them is differentiated by its side group. That's how we know what each one is. And the side group is really important as well because it determines the structure and the function of the amino acid as well. So the amino group, acid group, carbon, hydrogen is all the same for all amino acids, but then the one thing that they have different is what is in their side group. Now, I mentioned that we have 20 different amino acids, but those amino acids can also be divided up into essential and non-essential amino acids. The meaning of essential is that it is required for you to get it from your diet. It is essential that you get it from your diet because your body can't make enough of this on its own. Non-essential is not so essential for you to get it from your diet because your body can actually make enough of this on its own. This is true for any nutrient that is referred to as essential or non-essential. When we learn about different vitamins, for example, even different fatty acids can be labeled as essential and non-essential. And all that is telling us is whether the body can make sufficient amounts of this on its own, or whether it is essential that you get the full amount from your diet because your body cannot make enough. So some of them are essential amino acids and some are considered non-essential. Now, this here is showing us a variety of different amino acids. You don't need to memorize the structure, but what I do want you to notice is that the side chain can really vary. You'll notice that the one on the far left is very simple. It only has a hydrogen as its side chain, and then the one on the far right is a lot more complex with even a ring structure attached. Now, when we use these building blocks of amino acids to create different protein structures, the first thing that we can do is link two of them together. And when we link two of them together, we call them a dipeptide. Now, the way we join two things together, remember, is condensation, which is making water. So that's what you see happening here again. We create water through the condensation process, and that allows us to link the two amino acids together. So when we have two amino acids, we call it a dipeptide. So the bond that you see there that is holding the two together in red, in the picture on the right-hand side, you see a red bond, that is referred to as a peptide bond. So a peptide bond is actually what holds two amino acids together. So that's what the structure is named after, dipeptide. Now, when we link a bunch of peptides together, we call them polypeptides. And usually these polypeptides will be large enough to be referred to as proteins. And protein can take a variety of different structures. We have four main structures: primary, secondary, tertiary, and quaternary. And we're going to go ahead and take a look at what each of these look like. So here you see on the far left-hand side, we have the simplest and very first protein structure, which is the primary structure. This is when you have a string of amino acids attached together, very simple. Now, with the secondary structure, here we're looking at the same chain that we were looking at in primary, but now it's twisted into this helix shape. Now, the reason that this happens is because, remember, each amino acid has a different side chain, which means that the side chain can be positively charged, it can be negatively charged, depending on what it is. Now, because of that, since different amino acids are linked together, the positive and negative side chains of these different amino acids are going to be attracting or deterring each other. And based off of that, they are going to cause this change in shape where the structure begins to twist. So the secondary structure happens as a result of the difference in charge of the different side groups. Now, with the tertiary structure, the third one, this is when you have that same side chain or that same chain of polypeptides that was in the helix form, now folded all over itself into this big jumbled mess. And this is because those side chains can also be hydrophobic or hydrophilic. And the ones that are hydrophobic are going to want to try to hide towards the center of the structure. And so the structure starts flipping and folding over itself. The last one we have is the quaternary structure, which is when more than one polypeptide joins together. So the ones that we've been discussing so far all just involved one chain. When we have more than one polypeptide chain, that's when we call it a quaternary structure. Now, in this picture, it shows you something a little bit different. You see these polypeptide chains, and you see that they are held together by these bridges that say S. These bridges are what we call disulfide bridges. They're made of 2 units of sulfur, and they are used to give extra structure to a protein. This isn't available in all protein structures, but some proteins will have sulfur as part of their structure because they need these bridges. This is another example of what a protein could look like. Over here we have the protein hemoglobin. And one thing that you'll see is we have the folded up structure, which we call tertiary. But since we have 4 different chains that are all attached to each other in some way, it is actually called the quaternary structure. The structure as a whole is quaternary. Now that we have down the different types of polypeptide chains, different protein structures, we can go ahead and take a look at the digestion part of protein. Now, what we've learned so far is that since protein is usually in this jumbled up mess, the enzymes can't really access all parts of the protein properly in order to break it down. So we want to uncoil or basically untwist, untangle up the protein, and we do that by using acid or heat. That's going to deactivate and denature the protein so that it's more accessible to enzymes. Now, if you remember, the place where we have acid is in our stomach, and so that's why initially, starting off from in the mouth, we didn't have protein digestion. We had to wait until we reached part of the digestive system that had acid so that it could be denatured, and that was in the stomach. We used hydrochloric acid. After hydrochloric acid denatured the protein, now it is ready to be broken down by the enzyme. The enzyme that we mentioned was the enzyme pepsin. Pepsin is what is responsible for breaking down protein while it's in the stomach. But pepsin actually doesn't come in its active form. It starts off as pepsinogen, which isn't active, and it needs to be activated into pepsin. And that's where hydrochloric acid helps protein out again. Hydrochloric acid, or HCl, activates pepsinogen into pepsin. So HCl is very important for the digestion of protein. First, it helps to denature the protein so that it is accessible to the enzyme. Then it activates the enzyme from pepsinogen to pepsin. Now, after this, the protein is going to move from the stomach into the small intestine. And then in the small intestine, we're going to have the final digestion. If you remember, we had the pancreas releasing pancreatic enzymes into the small intestine, and part of those pancreatic enzymes were enzymes responsible for protein. This includes what we call peptidase enzymes, which is going to break down the peptide bonds. Now, after the protein has been digested, it can go ahead and be absorbed into the cells of the small intestine. And once it's absorbed into the small intestine, the blood will go ahead and pick it up, and then it can be used for a variety of different things. Now, the priority for those amino acids is to come together to build different protein structures in the body. Protein makes lots of different things in the body, including things like your cells, your muscle, other tissue, hormones, and so all of these structures need amino acids to be made. So that's usually what those amino acids are busy doing. If there are any amino acids that aren't being used currently, some of them will go over to the liver and be stored away in this very tiny space called the amino acid pool. So there is a little space for free individual amino acids. Now, for the protein structures that we're going to make, we're going to go through a process called protein synthesis, where we're going to go ahead and build different protein structures by using the information that we get from DNA. So DNA is going to basically give us the information that we need for how to build whichever protein structure we are looking to build. Whether it's a cell, a hormone, DNA is going to be what tells us which amino acids we need and which order we need to put them in to make that specific structure. There are a few other steps that we go through after that, but I'm going to go ahead and show it to you in the diagram. So here this is showing us the steps in action that were listed in the previous slide. So we start off at the very top. You see the DNA helix. This DNA contains information about how to make different protein structures. It's going to tell the body exactly which amino acids we need and which order to put them in so that the structure can be formed properly. So the first thing that we're going to do is we're going to go ahead and make a copy of this genetic information. And that copy is called mRNA. So we are going to make a copy of DNA. That copy is called mRNA. And this process of making the copy we call transcription. So that's step one. Step one is transcription, which is making a copy of DNA. And that copy is called mRNA or messenger RNA. The reason it is called messenger RNA is because it's actually going to take this information over to our protein-making factories, which are the ribosomes. The ribosomes are where we build our proteins, and so they're the ones who need the information on how to build the protein structure. So the messenger or mRNA is going to relay the information to the ribosome. And that is our step 2 that we call translation. Now, after that, the ribosomes now know exactly which amino acids they need and which order they need to put them in. So they're going to go ahead and start calling out on these amino acids. Now, we have some other helpers that we call tRNA or transfer RNA that are going to be waiting around to help out in this process. These are the little green things that you see hanging around with different colors on top of them. Now, the different colors signify different amino acids, and what they're doing is they're waiting around until they can deposit the amino acid that they are responsible for. Each tRNA is responsible for a specific amino acid. For example, the one that is carrying the red amino acid will always only carry a red one. So it's going to wait around until the ribosomes say they need a red amino acid, then it's going to come in and go ahead and drop it off. Then it'll go over to the amino acid pool and pick up another red one so that the next time ribosomes call for red, it's ready to go. And that's what's going to keep happening. The ribosomes call on the different amino acids, transfer RNA goes over to pick them up and put them in place. And we're going to keep doing this until we have a string of amino acids long enough and complete to build our protein structure. Now, sometimes there is an error in this process, and that's when we can develop things such as sickle cell anemia, which you see in this picture over here. Now, on the right-hand side, you see what a normal red blood cell should look like, and on the left-hand side, you see where there was an error in the making of this cell, and it's a lot different structure than what it should look like. The reason this happens is because the people who develop this have some kind of genetic defect where the DNA actually is wrong when it tells the mRNA how to make the protein structure. This is what we call a sequencing error. It's when the DNA will say, for example, put the red amino acid before blue when it's really supposed to be blue before red. So the amino acid sequence is wrong somehow. And because of this, remember, all the processes that happen after that are based off of the directions of DNA. So if DNA has something wrong, the way that we build the protein is going to be wrong. And we can result in a protein structure that is not what it's supposed to be. And that's what we see here. Because there was a genetic error in the DNA that told our body to make the red blood cell using a different sequence of amino acids, the red blood cell actually came out looking a lot different. Now here you see why this is so important, because at the top you have the red blood cells that are normally developed that can flow really freely through the blood vessels. But the ones on the bottom, since they have this odd shape, they can actually get stuck. They can lodge themselves in the different areas of the blood vessels and actually get stuck and block blood flow. Now, we did say that the main role for proteins is going to be building structures in the body. So that's going to be the main priority. Lots of the structures in the body are made of protein. This isn't just your muscles, it includes tissues, cells, lots of different things. We also have things like enzymes and hormones that are also protein structures. We even have transporters that carry nutrients, as well as transporters for good things going in and out of the cells that are also made of protein. This is an example of a transporter that is a protein. This is what we call the sodium-potassium pump. It is a transporter that is lodged into the cell membrane, and it opens up to take in sodium and release it to the other side, and then it'll open up to the other side to take in potassium to spit it out to the inside. And so this is basically a transporter that just interchanges things inside and outside of the cell. And this is just an example of one of the many things that protein can be used for. Now, we already know that in situations where we don't have enough carbohydrates, the body can use ketones for the most part, which are made out of fat. But if you remember, we said that there are two parts of our body, our brain and the red blood cells, that can't run off of anything but glucose. And so in situations where we are running low on carbohydrates, we have to find a way to make glucose if we're not giving it to our body. in order for our body to continue to function and fuel the brain. And in those situations, we said that we go through a process called gluconeogenesis. If you remember from the carbohydrates chapter, that was making carbohydrates, making glucose from a non-carbohydrate source, primarily protein. So that's something that protein can be used for in those desperate situations, but it's obviously not something we want to do. We want to try to spare protein as much as possible. We don't want to make it into glucose and use it for energy, because if we do, we're going to be breaking down our muscles and tissues and using that protein to convert it into glucose, because we don't have storage that we can take from. So that's only going to be something that we're going to do in desperate situations. Another thing that we can do with fat, or that we can do with protein, is convert it into fat. And this is going to be when we have too much protein. Again, we don't have storage space for protein. So if you eat more than what your body needs, then your body is going to convert it to fat because it can store unlimited amounts of fat. So it doesn't have anywhere to store it as protein, but if it converts it to fat, there's always room in our body for fat storage. And so that's something that can happen as well. But in these situations when we're going to convert protein to either glucose or fat, the first thing that we need to do is take off the nitrogen group. Remember, our carbohydrates and our fats are made of carbon, hydrogen, and oxygen. Protein also includes nitrogen. So if we're going to convert protein to carbohydrates or fat, then that means we need to remove the nitrogen first because they don't contain nitrogen in them. So that's the very first thing that we do, is removing the nitrogen group, and that is what we call deamination. Deaminating an amino acid is removing the nitrogen group, and that's what we're going to do in situations where we're going to turn it into fat or carbohydrates, because those two substances, those two nutrients, do not contain nitrogen in them. Now this here is showing us how the deamination process happens. So we have our complete amino acid, and remember we said we're going to remove the amino group. So the amino group is NH2. We want to get rid of that because we can't have nitrogen if we are making it into carbohydrates or fat. So we want to get rid of that nitrogen group, but along with it goes the hydrogen as well. So we have that individual hydrogen that's going to join NH2 and break off, making NH3. And NH3 is actually ammonia. So when we deaminate, we create ammonia, and then the leftover structure that we have, that is missing the hydrogen and missing the amine group, that is what we call a keto acid. So a keto acid is the result of deamination. When we deaminate, we are releasing ammonia, and we're left over with a structure called keto acid. Those are the two byproducts. Now, one thing that we saw is that when we deaminate, we're left over with ammonia and a keto acid. And the problem is that ammonia is actually something that is toxic if it builds up in our body. So that's not something that we want to happen. So the way that our body prevents ammonia toxicity is by the liver converting ammonia into a substance called urea, because urea is a lot less toxic than ammonia is. Now, after the liver has converted ammonia into urea, then the kidney is going to go ahead and filter the urea into your urine so that when you use the restroom, you are releasing that urea. Now, let's say that we looked at somebody's bloodstream and they had a buildup of ammonia. That would tell us that there is some kind of liver dysfunction going on, because the liver should be converting ammonia into urea. If we instead have a buildup of ammonia, that means the liver didn't do its job of converting it into urea. There's something wrong with the liver. Now, if we found a buildup of urea in the blood, that would tell us that there's something wrong with the kidney, because the kidney is supposed to filter it into the urine and get rid of it. But if it's instead staying in the bloodstream and building up, then the kidney is not doing its job. This here is showing us just an image of how this happens. So we have the amino acids in our diet. We are going to deaminate them, which leaves us with the byproduct ammonia. Ammonia is going to be converted to urea in the liver, and then it's going to go over to the kidney, where the kidney will put it into your urine, and then it will be excreted out of your body. So we've seen one of the reasons why we would deaminate, which is when we want to convert protein into a structure that does not contain nitrogen, like our carbohydrates or fat. Those structures do not contain nitrogen, so if we wanted to turn protein into one of those, we would first need to remove nitrogen off of it. Now, we also have another situation where we would deaminate, and that is when our body needed to make non-essential amino acids. Remember, essential are the ones that you need to get from your diet because your body can't make them. Non-essential are the ones that your body can make for you if you didn't get enough. So in a situation where your body had to make those non-essential amino acids for you, we would also start off with deamination. So we're going to go ahead and take a look at that process of making our non-essential amino acids and how deamination plays a role in that. So I have here for us a green amino acid. I'm just labeling them based off of their side group. This one has a green side group, so it will be our green amino acid. And let's say that we already had enough of the green amino acid in our body, and then we went ahead and ate a meal that had more green amino acids in it. So now we have all of these extra green amino acids that we don't need. But one thing that we didn't get was the white amino acid. Maybe we were lacking in the white amino acid and our diet didn't provide us with that either. Luckily, the white amino acid is going to be non-essential. And so since it's non-essential, that means it's okay that we didn't get it from our diet because our body can go ahead and make it for us. When we say that the body can make it for us, what that usually means is that the body at least has enough components to put together the keto acid form of the amino acid that we need. So in this situation, we need the white amino acid. It's non-essential. That means our body can go ahead and make it for us. It at least has enough components to put together a keto acid of that white amino acid. So here we have the keto acid form. So it is, again, the same as an amino acid structure, but without the amine group, and that's why we call it a keto acid. So in this situation, our body has put together the majority of the amino acid structure, but it's still missing that important amine group that turns it into an amino acid as opposed to a keto acid. Now, luckily, we have a bunch of extra green amino acids that we don't need, so we can go ahead and break those apart and basically use those for spare parts to help us build our white amino acids. So in this situation, we would also be deaminating. Remember, deaminating is when we remove the amine group. But the reason we would deaminate here is because we want to use this green amino acid for spares. We want to use it for spare parts. We want to break off its parts and use it to build the amino acid that we need. So remember, with deamination, we remove the hydrogen and we remove the amine group. Those two break off together in our deamination process, and together they make ammonia. And remember, after we deaminate, the structure that we're left over with can no longer be called an amino acid because it doesn't have the amine group. So instead, what we call it now is a keto acid. So again, we deaminated our green amino acid. That gave us ammonia, and the structure that was left over was a keto acid. Now, normally, once we make ammonia, we said we would convert it to urea because ammonia is toxic and we want to go ahead and kick that out of our body. We want to release it out of our system. Now, in this case, we deaminated because we wanted that ammonia. We wanted that amine group because we wanted to use it to build our white amino acid. So we don't want to convert it to urea. We don't want to kick it out of our body. So we're not going to do that process. Instead, we're going to hold on to the ammonia and we're actually going to transfer it over to our keto acid. So the ammonia is NH3. We can go ahead and take one of the hydrogens, put it on this side, then take our remaining NH2, add it on the other side, and now if you take a look at this structure, it's actually a full amino acid. So it was only called a keto acid before because it didn't have its amine group. Now that the amine group is there, it's no longer a keto acid, now it is a fully formed amino acid. So basically what we've done here is we were able to build the amino acid that we were lacking in by taking the amino acid that we had excess of, deaminating it so that we could use it for spare parts, and then using those spare parts, the ammonia, to transfer them over to the structure that we were lacking, and we were able to put that together and make a full amino acid out of it. This process over here is what we call transamination. So remember, deamination is removing the amine group. Transamination is transferring the amine group. So again, this is another situation where we would deaminate, but after we deaminate here, we're not converting it to urea and we're not kicking it out of our body. We're holding on to our ammonia so that we can transfer it over and help build the amino acids that we were lacking in. All right, now we're going to go ahead and take a look at the recommendation for protein in terms of your diet, how much we want to consume, and how we want to determine if we're consuming the right type of protein. Now, starting off with the right type, two things that we want to look at are digestibility and the amino acid composition. Those are the two things that are going to determine how high quality a protein is. If a protein is easily digestible and it contains all of the essential amino acids, then it is considered high quality protein. Now, animal protein tends to be more easily digestible than plant protein because the plant protein contains a bunch of fiber usually. And so that is the first thing that makes animal protein higher quality, is it's easier to digest. The other thing that makes animal protein high quality is that it always contains all of the essential amino acids. So your plant protein usually will be missing some of the essential amino acids, but usually animal protein will have all of them. So animal protein really is the higher quality protein. That doesn't mean that if you are vegetarian or vegan that you're not going to be able to get high quality protein. You can, but there are some things that you need to consider in order to make sure that you are getting high quality protein in your diet. Now, another thing to consider is that we want to make sure that we are getting complete proteins at the very least. And complete protein is part of the quality check that we were discussing, which was, do we have all of the essential amino acids? If you are vegan or vegetarian, this is something that you can make sure to do, even though you can't do anything about the digestibility of plant protein. You can at least make sure that you are ending up with a complete protein, which means you're getting all of the essential amino acids. If it's missing some of the essential amino acids, we call it an incomplete protein. And that is something that we can also do something about in order to have a complete protein source in a meal, and that is what we call complementary protein. So looking at what complementary proteins are, basically you are taking different protein sources that are missing some of the essential amino acids and combining them with other sources that can fill in those blanks. So for example, over here we have legumes or the beans that are missing the last two amino acids, and then we have your grains, the rice, that are missing the first two, but they contain the last two. When you eat these together, the beans and the rice, you end up with all of the essential amino acids, and so now you have a complete protein source. So when we say something is a complementary protein source, that means that we have put together two different proteins that fill in each other's blanks when it comes to the essential amino acids that they're missing. Now, as for the actual recommendations that we want to follow when we are deciding how much to eat in the day, we already learned the AMDR is 10 to 35% of your daily calories, but if you want to do it based off of weight, it is 0.8 grams per kilogram of your body weight. This is for the typical healthy adult. Now, most individuals actually get a lot more than this amount. Athletes do tend to need a little bit more, and we'll talk about that in more detail in the fitness chapter. But even that higher requirement for athletes, we still tend to get as much as an athlete needs, even for the individuals who aren't athletes. It's just a trend in the US where our protein intake is a lot higher than our needs. Now, because of the fact that we do tend to get too much protein, protein supplements are really not necessary, whether it be individual supplements or protein shakes. It really isn't necessary. And again, we'll look at this in more detail in the fitness chapter, but basically, even the typical individual who is not an athlete will usually get as much as an athlete needs. And then athletes, when they're trying to supplement with all these protein shakes, they are exceeding what their requirements are. And remember, when you exceed what your body needs, it will turn into fat. So for that reason, protein supplements are really not necessary. Now, on the other end of the spectrum, we have people who aren't getting enough protein. And we have two main types of diseases where there is protein deficiency, and those are marasmus and kwashiorkor. And let's go ahead and take a look at what those look like. So on the left-hand side, you have marasmus. That is one protein deficiency condition. And this is when the person is not only lacking protein, but they're also lacking calories as well. So their body is already not getting enough calories, so it's already breaking down its structures. But then to top it off, they don't have protein to maintain their own structures either. So their protein breakdown, their muscles, their tissues are going to break down at a much faster rate. Now, on the individual that you see on the right, this is someone with kwashiorkor, which is when they are maybe getting enough calories but not enough protein. This tends to happen in a lot of developing countries because protein tends to be something that is more costly, and so a lot of times they're living off of carbohydrates. And in that situation, they're still going to have loss of their tissues, but it is going to be at somewhat of a slower rate because they do at least have enough calories. But one thing that you'll notice, if you look at this child, the arms are very, very thin, but then the stomach and the legs look bigger and more full. This look that there is in the stomach and the legs is actually what we call edema, or collection of fluid. It is basically when the balance of fluid in the body becomes disturbed because of a lack of protein. One of the roles of protein is to keep your fluid balance in check. And without protein, you can start having a lot of fluid accumulating in different parts of your body, especially the lower half, and collecting right underneath your skin. So this individual with kwashiorkor is actually losing a lot of its tissue and muscles, but it doesn't look like it because they have a lot of fluid collection, which is its own complication in itself. Now, the measure of the protein balance is also sometimes referred to as nitrogen balance. And this is referring to how much nitrogen is going into your body in the form of protein and how much is leaving your body. So think of it in a way as your protein balance. Now, usually you should, if you are a healthy individual, you should be at normal balance or what is called zero balance, but there are certain situations where people might be in positive or negative balance. Positive balance is when your intake of protein is greater than your output. So you're putting a bunch of protein into your body, but not all of it is being released, if any at all. Now, if you think about it, this is going to be in times when your body has an extra need for protein. So, for example, if you are going through a growth spurt or it's a child that's growing, still in its growth process, they are going to be using up every little bit of protein that's put into their body. So we're putting in a large intake, but then we're not having much, if any, output because the body has used it all. It's not releasing it. And so our intake remains higher than our output. Another example is if we have a pregnant woman. A pregnant woman is going to use up that protein for her own needs and then for the child's needs as well. And so she'll be putting in a bunch of protein into her body, but her body's not going to release it. It's going to go ahead and use it for its needs. And so what we have left over, or what is released from the body, if any, is going to be very minimal. So think about it as it is positive balance anytime your body has an extra need for protein, anytime you put protein into your body and your body is going to use it all up instead of releasing it. So positive balance, more intake than output. Now we have negative balance on the other end of the spectrum, and that's going to be usually when you have some kind of breakdown of protein in your body. So, for example, let's say that we have someone who is starving, someone who's not eating. They're not putting much protein into their body, but they have a bunch of protein output. And this protein output is from all the muscle breakdown, the tissue breakdown, because their body is starving. So negative balance is when your intake is less than your output. So again, back to the starving person. They're not putting in much protein, but because of that, their body is breaking down its own protein structures in order for this person to survive, since it's not getting enough food. So their output would obviously be greater than their input. Another situation is going to be, for example, if you have some kind of cut, let's say, or a wound, a burn. The breakdown of that skin in the cut, the wound, the burn, whatever it is going to be part of the output. And so the output is going to be greater than the input. Now, there are times where, let's say you're recovering from this wound or this burn, whatever it may be, the protein that you put into your body is going to be used to rebuild the skin wherever you had your wound or your burn. And so in that situation, when you're recovering from an injury, it is going to be positive balance because your body's using up that protein to help you recover. But while you have an active burn, active breakdown of your skin, you are releasing more protein than you put into your body, because besides what you put into your body, you also have breakdown of your own structures. So negative balance is always going to be when you have some kind of protein breakdown in your body.